Hello, I am Eugene Rau from Timișoara city, Romania, and I was interested in how uh, magic of life is working. So I entered deep in this field, and my exposure is entitled "How Life Biochemistry, despite being extremely complex, is self-regulated and so coordinated." And I say, let's admit that I understand the formation of increasingly complex molecules in the universe from simple elements. Then, in the chemistry of the stellar environment, the formation of organic precursors up to abiogenesis. But how life appeared, what makes homeostasis exist, not only at the level of organism, but also at the molecular level, I no longer understand. That is, I do not understand how millions of substances and reactions take place as if they were coordinated when the simplest would be to take place disorderly. Not to mention the ultimate example, the genetic DNA mechanism. What kind of explanation would there be? And I did not know the place to find, and asking the AI assistant Gemini, which is which is responding. That's an excellent question that touches on the most fundamental aspects of life's organization: emergent order and self-organization. The key to understanding how millions of reactions can appear coordinated instead of disorderly lies in the principles of non-equilibrium thermodynamics and molecular control mechanisms that evolved to maintain homeostasis. Coordination vis-a disorder, the role of non-equilibrium. The reason life doesn't simply descend into chemical disorder, which would be the simplest state according to classical thermodynamics, is that living systems are open, non-equilibrium systems. Classical thermodynamics, equilibrium, the simplest state where energy is evenly distributed and no useful work can be done, is the highest entropy or disorder. Chemical equilibrium. A closed system naturally moves toward this state. Non-equilibrium thermodynamics. Open systems. Living cells are open systems. They constantly take in energy, e.g., nutrients, sunlight, and matter, and they constantly release waste and heat. This continuous throughput of energy maintains the system far from equilibrium. Dissipative structures. When open systems are maintained far from equilibrium, they can spontaneously form dissipative structures, highly ordered patterns or cycles that consume energy to maintain their order, e.g., a vortex in a sink or the metabolic cycles in a cell. Life, in this view, is a grand self-sustaining dissipative structure. Homeostasis and molecular control. Homeostasis is the process of maintaining a stable internal environment despite external changes. At a molecular level, this is achieved through sophisticated control mechanisms that dictate when, where, and how quickly reactions occur. This is what provides the appearance of coordination. 1. Enzymes and catalysis. Enzymes are the primary coordinators. They are highly specific protein catalysts that selectivity. Each enzyme typically catalyzes only one or a few specific reactions. This ensures that only the necessary reactions happen at any given moment, preventing a chaotic general mix of all possible reactions. Regulation. Enzyme activity can be turned on, activated, or off, inhibited by other molecules. This allows the cell to instantly adjust the reaction rates based on its current needs. E.g., if a product builds up, it may inhibit the enzyme that made it, a process called feedback inhibition. 2. Compartmentalisation. Compartmentalisation. The cell itself is not a simple bag of chemicals. Membranes, like the cell membrane, and membranes creating organelles, like mitochondria, create compartments. Spatial separation. Specific sets of reactions, e.g., glycolysis, fatty acid synthesis, are isolated in different compartments. This prevents unwanted interactions and ensures that the right concentration of reactants is present for a specific pathway. 3. Allosteric regulation and signaling. Many key enzymes have allosteric sites, binding locations separate from the active site. A regulatory molecule binding to the allosteric site changes the enzyme's shape, instantly turning its activity up or down. This forms the basis of highly sensitive signaling pathways like signal transduction cascades that allow the cell to quickly coordinate its entire metabolism in response to an external signal, e.g., a hormone. The genetic DNA mechanism. The genetic mechanism, DNA, RNA, and protein synthesis, is the ultimate blueprint and control center that ensures the coordination continues across generations and that the necessary molecular machinery, that enzymes and structural proteins, is constantly and correctly produced. Instruction set. DNA is a stable, archived set of instructions for making all the necessary coordinated components: enzymes, regulatory proteins, structural elements. Central dogma. The flow of information, DNA, RNA to protein, is a highly controlled process. When the cell needs a specific coordinated enzyme, the corresponding gene is expressed. copied into mRNA and then translated into a protein. Transcriptional regulation. This is the most complex layer of control. Specific regulatory proteins, called transcription factors, bind the DNA to turn genes on or off. A massive coordination mechanism ensures that, for instance, all the enzymes needed for an entire process, like sugar breakdown, are produced simultaneously and only when sugar is available. In summary, the coordinated nature of life is not a mystery but an emergent property of a complex, energy-driven, self-regulating chemical system. The simplicity of this order is overcome by the continuous input of energy and the highly specific, tightly regulated control mechanisms inherent in enzymes and the genetic code. Now I know a little more that I knew before. Thank you very much. Bye.